Thank you very much. Well, we are going to talk a little bit about our last studies over the last 3 years, trying to promote some new strategies that eventually can be translatable for ALS and other motor neuron diseases. As you may know, ALS is a neurodegenerative disorder primarily affecting upper and lower motor neurons and also their muscles, and resulting in progressive muscle paralysis, in most cases with early death in a few years. And as for other neurodegenerative diseases we have been talking about, there is no effective treatment, with, in most cases, only palliative care being available to prolong a little bit the quality of the short life of the patients. The typical picture now is changing. Probably in a few years these figures will be changing, but the classical is that there is sporadic ALS, in most cases, of unknown origin, etiology, and 5–10% with familial origin related to mutations in several genes that focus or promote the secretion of different proteins. There is some new findings about 2 years ago about mutations in the chromosome 9 region that will probably is going to be present in a lot of patients, 20–25% of the patients in some cases, and that probably will be changing a little bit the face of this picture. Based on the appearance on the existence of familial cases, and one of the most common ones, accounting for about 20% of this familial, is the mutations that are affecting the gene encoding for the copper zinc superoxide dismutase 1. Okay, and based on this mutation, the transgenic mice have been developed in which they are made to overexpress the human mutated SOD gene. Okay. This model is presently more relevant not only for the short percentage of familial ALS cases. In alterations in SOD1 protein and function have been reported in a lot of sporadic patients, so that the physiopathology of these mice may be relevant not only for the direct familial cases. These mice, and particularly the most common one, that is the G93A, recapitulates most of the pictures of the characteristics in the clinic for ALS. The mice develop well normally, motor behavior is correct until about 3 months of age, when they start to show some abnormalities in locomotion or in the rotor test, and that develops to fast paralysis, so concluding in death about between the 4th and the 5th months of age. Okay. The motor neuron death is also present, more or less. Loss of motor neurons in the spinal cord is coincident with the starting of paresthesia and is declining over time. So based on this model, and also with some correlates to clinical material, the picture of the pathogenetic mechanisms probably related to the development of ALS and other motor neuron diseases have become to be a very complex picture. Okay? There are a lot of abnormalities depicted here that may play a role. Excitotoxicity is one of the most relevantly work, but also abnormalities in the metabolism of the motor neurons, like developing endoplasmic reticulum stress, mitochondrial dysfunction and oxidative stress, abnormalities in the axonal transport, some potential synaptic vesicle defects or abnormalities in the neuromuscular junction relationship, and also some abnormalities relating non-neuronal cells present in the spinal cord in the brain, like the astrocytes and the microglial cells, that may become less effective, for example, taking out glutamate in the area, or also releasing a lot of protoxic effects that may affect also motor neuron. So this picture is quite complex. No single cause will probably be related to the disease. That has been made that several, we can say a lot of therapies, a lot of studies in the animal models that showed some benefit, partial benefit for the animals, have failed to report any significant benefit in clinical trials. So there is a lot of debate on the usefulness of the animal models because not much is coming for the clinical application. Okay? There's not only the molecular mechanisms, but also the cell targets are multiple here. Certainly there is involvement in ALS of the lower motor neurons, spinal cord and brainstem, the upper motor neuron, the brain cortex, but also the glial cells, as I mentioned, the inflammatory cells, T cells, and also the skeletal muscle has been involved. So the targets are molecularly multiple, cellularly multiple. So that following this way of thinking, probably therapies that may target different mechanisms of action could be more prone to provide benefit than therapies that are focusing on a single mechanism. Or alternatively, combinatory treatments seem to be the next future to go, okay, so that we can target different of the pathways that are affected. So that I will try to summarize our work with 3 different studies that are in different stages of production and focusing on new developments, new treatments that may be multi-target agents, multi-target drugs. The first one, I presented some preliminary initial studies about 2 years ago in this forum, was trying to target the sigma-1 receptor. Sigma-1 receptor, to be brief, is ubiquitously present in the nervous system, but particularly has high expression in the motor neurons of the spinal cord in relation with the endoplasmic reticulum and the postsynaptic sites. Okay? Some mutations in the sigma-1 receptor gene have been found associated with some familial cases of ALS, so that could be a relationship. There are quite a number of mechanisms targeting sigma-1 receptors, so that agonists, that may be drugs that bind to the sigma-1 receptor and promote its activity, can be preventing apoptotic neuronal death. It reduces calcium influx through NMDA receptor and other calcium channels, are able to reduce the activation of INOs and also microglial inflammatory activities. So different pathways that may be neuroprotective conducted by the activation of the sigma-1 receptor. And indeed, neuroprotective effects have been found in vitro and in vivo models of ischemia, the cerebral stroke, and also we had some data of protections of motor neurons under excitotoxicity insults in vitro. Okay? So based on these previous findings, we went to make a study on these animals, the SOD1 G93A mouse model, following the following design. We started the study about 8 weeks of age. 8 weeks is a presymptomatic phase when animals walk normally. They don't show abnormalities in the rotor rod. But by applying conventional electrophysiological tests, we can diagnose the mice inequivocally. They have clear abnormalities. So we could apply to an early diagnosis stage of the patient. At that time, we started treatment in one group with PRE-084, that's a sigma receptor 1 agonist. Another parallel group was treated with the same PRE plus BD1063, that is an antagonist, just to block the effect and prove that the potential benefits of PRE were mediated by the sigma receptor, with a control group and a wild-type group of animals. By every 2 weeks, we performed electrophysiological and locomotion tests to see the performance of the animals. And then by 16 weeks, 4 months of age, when the animals start to have clear symptoms, we eliminated a group of animals to obtain samples for histology. And another subsets of animals were followed until death, so to measure the survival of these mice. That is one of the standard goals in this type of research. To make briefly the results, the sigma-1 receptor agonist PRE really induced a clear improvement in the functional state of the animals. The compound muscle action potential amplitude for tibialis anterior or the plantar interossei muscle were clearly prevented. The decline in the control animals was clearly prevented in the treated animals, whereas treatment with PRE plus the antagonist really eliminated the positive effects. This effect was present not only in female mice, but also in males that usually show a slightly worse progression of the disease. So from a functional point of view, they were much better, and the rotor rod test also indicated that they were prolonging the capacity for locomotion. The histology, ventral horn, you can see that there is a marked decrease in these large alpha motor neurons in the lateral ventral horn. In control animals, it's about 40–35%, a little bit worse in the males, okay, the number of motor neurons present. And you can see that by the treatment with PRE, there's about double number of motor neurons surviving at the time of sacrifice, 16 weeks of age. Combining this picture, microglial cells, I don't know if you could turn off, si podéis apagar la luz de aquí delante, porque se ve muy mal esta pantalla. There is a strong macroglial reactivity surrounding the motor neurons in the ventral horn, And this microglial reactivity that was also measured as percentage of immunoreactivity was clearly reduced by the treatment with the PRE, with the sigma agonist. Okay. Survival was significantly increased by about more than 10% of the time. See that females or males die about 125 days of average, and with the treatment, it was prolonged by 15% of days for the animals. So all these indicate a good prognosis, a good possibilities for this treatment. Then we moved to treat another group of animals under the same regime with the same PRE, 084 drug, but starting at a more advanced stage of the disease, 12 weeks of age, when there is really a decline in the locomotor performance and also in the amplitude of the muscle action potentials. We again found that the protection, so the decline was stopped for the amplitude of the action potentials, and the animals continue to walk at certain speeds that the not treated animals were unable to do. There was still some preservation of the motor neurons in the spinal cord, but less than when we started earlier the treatment. So that really this points to the need for early diagnosis, even pre-symptomatic diagnosis in the patients, and the eventually very early treatment to protect the loss of motor neurons. Okay, this following study was focused on the use of resveratrol. And the interest in of our group was prompted by two facts. One was related to CIBERNED, because in one of the last meetings we talked with another group of CIBERNED, Toni Camins and Mercè Pallàs from the University of Barcelona. They have been using resveratrol in Alzheimer's models, and found that good effects and increases in lifespans very significantly in this model. And there were also a few publications, and one recent one was showing that in vitro, in motor neurons in vitro, the addition of resveratrol was helping the neurons to survive in a, let's say, motor neuron-like cell model. Therefore, we started the study using pointing out that resveratrol is a multi-target drug. Okay? It acts primarily by activating sirtuin-1 or AMP kinase. And through these mechanisms, it may improve genome instability, reduces oxidative stress, reactive species secretion, reduces inflammatory responses, increases mitochondrial biogenesis. So this has been pointed that there are a variety of positive effects in different studies in other diseases, Alzheimer's and other type of diseases that may affect the nervous system. Following the same design, so we administered from the 8th week of age the animals with resveratrol. Resveratrol was added in the diet, so it was an easy sustained administration for the animals, at a average dose of 16 milligrams kilo a day. 2 weekly follow-up, and some sampling for immunohistochemistry and molecular biology at 16 weeks, and other animals were followed up until death for survival analysis. The resveratrol the administration in the diet clearly delayed the disease onset and improved locomotor performance in these animals. As you can see, the disease onset is marked by the starting fall down in the rotarod locomotion test. So that you can see that was really prevented or delayed in about 1 to 2 weeks in females and male mice. And the rotarod performance was always better during the follow-up, during the 16 weeks follow-up in both sexes treated with resveratrol with respect to the corresponding wild type, SOD1 controls. Certainly the wild types are sustained all the time at 180 seconds. For the electrophysiological tests that allow us better quantitation and mark differences, you can see that there was still a prevention of the decline of the muscle action potentials in the tibialis anterior muscle and also in the plantar muscle. But in this case, I want to point out that also we are performing regularly motor evoked potentials also in our mice, just to control for the upper motor neuron, the cortical motor neuron. And then in this case, for the first time in the literature, we have found that there is a prevention by a treatment in the functional loss of the upper motor neurons. So the amplitude of the motor evoked potentials at the percentage of animals that present recordable motor evoked potentials are significantly increased by the addition of resveratrol. So that points out to a potential two-side beneficial effect. This looks like the same slide, but this is not the same. This is referring to male mice, because we have to treat in these animals, have to evaluate both sexes to really be sure what's going on. But you can see that in males also the effect was significantly positive, and also for the evoked potentials. Resveratrol also extended SOD mice survival. In this case, our control survived. There is different batches of animals, so there are minor variations also in controls, about 135 maximum time. The median was about 132, and there was about 12% increase in the median survival rate. So that incorporating also an additional effect of the animals. When we look at the histology, again in the ventral horns, you could see that there was a marked loss in the control mice. Also, some of the present motor neurons, as you can see, they don't look very normal, so they could be atrophic and probably dysfunctional. And you can see a much better picture for the resveratrol animals. Okay? And when we quantified the number of motor neurons present at the lumbar sections of the spinal cord, you could see also that we almost double the number of motor neurons surviving, approaching the normals with the treatment with the resveratrol. All that indicates that this is a potential treatment that could be effective and could be easy to handle for translation, probably easier than the sigma-1 receptor agonist. Although there are sigma-1 binding drugs already in clinical assays. For example, there is an antagonist for neuropathic pain in phase 2 assay already. Resveratrol also reduces microglia, so it was also able to modulate the inflammatory response. And you can see that clearly the immunoreactivity for resveratrol was clearly reduced in these animals. Okay, whereas it did not affect astrogliosis. So the astrocytes labeled with GVAP look like the similar in the not treated and the treated SOD1 mice. To look for the potential effects that have been targeted by resveratrol, we look at the sirtuin expression. And as you can see, resveratrol was able to activate sirtuin-1 expression very highly in the spinal cord. And when we look at the immunohistochemistry, we found that the expression of sirtuin was really effectively apparent in the motor neurons. Okay? And also that this high expression of sirtuin was effective in controlling the pathway was proved by the decrease in the acetylation of p53, that is one of the targets of sirtuin with resveratrol treatment. Okay? So this was really the proof of concept that the resveratrol was doing what was expected on the pathway. Okay? Interestingly, in a cooperation, this study was made in the laboratory in Lleida of Dr. Manuel Portero. He looked at the respiratory chains because resveratrol has also some antioxidative effect. So we wanted to look at the mitochondrial function. Curiously, they found that all the 5 respiratory chain complexes were overexpressed, were hyperactivated in the SOD1 mice, probably indicating that the motor neurons that are surviving, this is coming from samples from only a small piece of the spinal cord, the ventral quadrant. So probably the motor neurons are suffering and are proactivating like a stressful response, all the respiratory chain complexes. And this was normalized for practically all the cases except for the complex 4 with the resveratrol treatment. Furthermore, by analyzing a little bit more the effect, that was evidence that was increased mitochondrial mass. And that was proved by fission and fusion immunohistochemistry on Western blots. That was an effect that was promoting mitochondrial biogenesis, so increase of mitochondria present at the motor neuron. So we had 2 targets potentially useful and targeting different actions around motor neurons. So we also wanted to look at other cells that are accompanying the motor neurons and that have been implicated in the pathophysiology of motor neuron diseases. Inflammation mostly including microglial cells, but also T-cell infiltration is a hallmark of the neuropathology in ALS samples and also present in the mouse models. Okay? Initially can be interpreted like a defensive mechanism. So these cells are trying to eliminate detritus and improving the survival of the remaining cells. But while later it has been proven that they contribute to accelerate the disease by itself. Probably the accumulation of the misfolded mutant SOD1 protein that is not also affecting sporadic cases, not only the transgenic cases, can be a relevant factors, making that the microglia are activated, are transferred from a resting protective type of phenotype to a more cytotoxic phenotype M1, and that can release reactive auxin species and pro-inflammatory cytokines, so that may help to deteriorate the potential life of the motor neurons. Okay? Indeed, the suppression of microglia by just anti-inflammatory drugs have not been proven of much benefit in the animal models, because probably we don't want to suppress all the potential effects of microglials. So the idea following this third study was that we wanted to be able to modulate, to regulate microglial proliferation and activation, and also try to see if we could make a predominant expression of the M2 neuroprotective phenotype microglia versus the M1. To start in this area of research, we focused on a compound, GW, we are going to tell, that specifically blocks the activity of the colony-stimulating factor 1 receptor. And by blocking this receptor, it affects inhibiting microglial proliferation, okay? And it may promote partially the expression of the M2 phenotype versus the M1. It has been proved good effects in other models like prion infection, prion disease animal models. So we, for the first time, attempted them in the SOD1 mice. Again, the design follows our typical design of a study, starting at 8 weeks, following animals to 16 weeks, and then for survival. And in this case, the electrophysiological effects are again very good. So the muscle action potential is prevented. The decline is half of the decline that is in the non-treated animals. Interestingly, the more distal plantar muscles have comparatively better percentage of recovery than with the previous 2 treatments. On the other hand, functional overall results with the rotarot or the digigate forced locomotion were not so well preserved. Okay? Indicating probably that we are acting through other mechanisms. It's not repeating again the same action. So probably we are touching on other sides, and these effects, the more that are relevant to all the motor unit, whereas the complete locomotion tests include a lot of other circuits, may point out to effect at the motor neuron and the periphery more than at the more central levels. Survival was also significantly prolonged with the GV treatment. Okay? This study is still in the kitchen, so please don't tell many people, because these modulators are being actively investigated by several groups. We had some analysis of the microglial population at these ages from samples and FACS analysis. And from this initial preliminary study, we can already see that our compound, the treatment with this compound, was effective, reducing the proliferation of microglial cells in animals at 16 weeks of age, very advanced at the stage. You could see previously some immunohistochemistry full of microglia. And the proliferation was reduced, and maybe a slight reduction of the activated percentage of microglia. So we are really affecting the microglial proliferation, and probably its direct and indirect effects on the motor neurons. So that, recovering again this first slide that I showed, we have now some compounds that target different areas of this complex image. Excitotoxicity and ER stress, probably acting through the sigma-1 receptor, the microglial cells with this GW modulator, mitochondrial dysfunction and oxidative stress with resveratrol. Probably the next step is going to be to make a thorough analysis, comparative analysis, and try to design combinatory drugs, combinatory treatment with these drugs, and hope that by this strategy we may come this year and show you better results. So just want to thank all the people in the group, particularly the persons who have been dealing and fighting with these animals, Renzo Mancuso. Okay? Jaume Del Valle also cooperated in the resveratrol study. Laura, Eva, Santos, Isaac, and Anna are helping also in the studies. And the cooperations with the group of Charo Osta in Zaragoza, with whom we have a continuous cooperation in these studies with the SOD1 animals, with Mercè Pallàs in Barcelona and CIBERNED, And the last study about microglia with the group in Southampton of Hugh Perry, and particularly Diego Gomez Nicola, who is now at Southampton working. And thank you very much. That's all.
Questions?
Okay, thank you. Xavier, one question. So, what about the effect of resveratrol in the wild type?
We didn't find any neurological difference or with respect to wild type animals.
No,
I mean, they may have better survival or better reduced oxidative stress. That may be effect, but we haven't looked at in detail at the wild type because the comparison are with the SOT1 model. So the point is, if you improve generally or you are improving specifically the mice, I think that for the electrophysiological studies could be impossible to know because the wild types at this age have no loss of amplitude or loss of function. They are very stable. They are very quite young mice at this age. So we have to look probably only at some molecular cascades to take something into account. We haven't looked at it in detail.
Okay.
One short question. Are you planning to combine the treatment of antimicroglial compound with resveratrol to potentiate both effects?
We haven't decided yet. We are planning another combinatory study, but it's still to be started. So I don't tell. Probably with combining cell therapy and one of these treatments, resveratrol. With the microglial modulation, we are in the phase that the samples are being characterized by immuno and some molecular biology, Western blot, etc., for the type of microglia, the production of cytokines, etc., and also in the cooperation with Southampton. So we have to have the overall picture and really find if it's important, the molecular effect, to sustain this functional effect. Eventually, the next step could be that, to combine the effects. Certainly, combination, it's not an easy solution. From being very naive, one thing, well, if I have improvement of 2 with one treatment or with 2 with others, if I put the 2 treatments, I will have 4. But we know from previous experience in other neural injury models that this is not always true. And sometimes you still remain with 2 because you are not really adding new effects. Also, the SOT1 models, like the ALS patients, are very complicated models because there are certain number and types of motor neurons are dying very early, earlier than the symptoms start, even earlier than the electrophysiology starts, because the motor neurons can compensate the loss of the initial motor neurons lost. The others can sprout and recover muscle territory. So our hypothesis is that there is some population of motor neurons that's going to be very, very difficult to protect. So they are going to fall very fast. So the potential is we have to preserve the others and make them to gain function, to be able to compensate the loss that we cannot avoid at the very early stages of pre-diagnostic diseases. So, this topic is also important to consider when trying to combine therapies, to know a little bit how they will be acting and if they will be exactly complementary. Certainly, I was interested in showing you all this picture, even the still to be finished studies, because if anyone has, promising compounds that may promote neuroprotection in other models, certainly the cooperation in CIBERNED will be always welcome for us.